The following is about the Nirenberg and Matthaei and the Nirenberg and Leader experiment about determining the coding dictionary. Up until this point, we understood that RNA coded for amino acids, but we had no idea which codons coded for which of the amino acids. So, the Nirenberg and Matthaei and Nirenberg and Leader both devised an experiment to help determine which of the codons coded for what. So Nirenberg and Matthaei came up with the first experiment, which is poly-U experiment, where they mixed poly-U in the cell-free extract in these test tubes where one of the amino acids was radioactive. They repeated this many, many times to figure out which amino acid the poly-U would react with. What they noticed was that the poly-U only reacted in the test tube where they had the radioactive phenylalanine. Nirenberg and Matthaei repeated this with a poly C, a poly A, and a poly G. What they saw after repeating it all the times was that the poly C reacted in the test tube with lysine, the poly A reacted with proline, and they could not get the poly G to work. At this point, we had figured out what 3 of the codons would code for, but we had no idea what the other 61 were. So Nirenberg worked with another scientist, Leader, where they took mRNAs of all the different combinations and they added it to a mixture of ribosomes and tRNAs. They would try to see if the mRNA would bind with the tRNA in the ribosome. If it did bind, then they would filter it through, and if they filtered it through and it binded, they were then able to take what had bound to the top and what had not fallen through, and they would look at it and evaluate it and figure out which amino acids and which of the mRNAs and tRNAs were left in the mixture. With this, they were able to figure out the other 61 codons. So, the conclusion of this experiment with Nirenberg and Matthaei and Nirenberg and Leader is we were able to decipher the coding dictionary, which is important for our understanding of the central dogma and understanding for which codons code for certain amino acids, because it is nearly universal and works in most organisms and cells.